My name is Greg Parr. I work for the USDA. I'm part of a 3-member group of the SLF cross-functional working group within our agency. The other 2 members are Aaron Otto and Matt Travis. I work for the science and technology part of our agency. I'll be moderating this session of the SLF research. So we're going to jump right into it and begin with this research will be focused on grapes, and there'll be 4 presenters, so they're divided up their time. That's why we don't have specific time points on there, but yes, I feel like it'll work out, and everybody will meet our deadline of 12:30. Okay, so the first presenter will be Dr. Greg Krycek, who's a research professor and extension tree fruit entomologist at Penn State, and he'll be talking on ovicides and exclusion. So are you all set, Dr. Krycek?
Good morning, everyone. It's a pleasure to be able to actually do it in person. I am right now in somewhere in Pennsylvania, in Blair County, at the grower meeting, and I'm talking from some kind of supply room, but I hope my internet connection will work for the next 20 minutes, so I can share with you some of the research that I did, my program, my lab did, working on spotted lanternfly. And I am part of the grape session, but the topics I'm going to cover are not necessarily only grapes related. So I know that everyone knows the biology of spotted lanternfly. I thought I will show this slide because I like it a lot. I believe all the photos on this slide are from my camera. However, the reason why I'm showing it is to emphasize that, you know, since we have only 1 generation of spotted lanternfly, breaking the cycle at any point, at any moment, actually is destroying the whole cohort of the spotted lanternfly. So being a fruit entomologist and working a lot with the fruit growers, I kind of concentrate on trying to understand what can we do against overwintering stage, which is overwintering egg masses. We started working on spotted lanternfly back in 2017. We ran a lot of different bioassays. We ran them in the lab, we ran them in the field. I will not be able to present all of the results. I hope to share with you some of them. One thing also to understand is that my lab is located in Adams County, Pennsylvania. And if you look at the distribution of spotted lanternfly, at least until today, Adams County is not under quarantine. We are surrounded by counties that have spotted lanternfly established population, but that's not the case for our area. As great as it is for the fruit growers around us, for us, it creates a big problem. You have to do all the research outside of the lab. We don't have the quarantine facility. So we are working in growers' offices. We are working in growers' shed, in the vineyards. But to get to any of our sites, it takes us more than 1 hour to drive to be able to do something. This is an example of our field lab, which is in Lancaster County. It's only 1 hour, 20 minutes away from us. Grower create the tables for us and let us use one of his barn, so we actually can run some research. We try to do it the best way possible. However, it is a barn and some limitations exist. We had the power there, so we can use incubator for keeping the constant temperature. We use a lot of cages, and well, we do what we can, trying to learn as much as we can about spotted lanternfly and what can be done to control those overwintering eggs. As I mentioned earlier, I'm not gonna talk about all the bioassays we did. We have a lot of them. I don't know the number of products we tested, but based on our experience, with other insect pests based on our understanding of the way how different pesticides, insecticides are working. We choose certain products and try to assess if in any way they are effective against spotted lanternfly overwintering eggs. In typical bioassays where we spray eggs directly and then put them into those incubators and then check the hatch, we found out that the best product that is out there that controls spotted lanternfly eggs almost perfectly is chlorpyrifos, which is the active ingredient of Lorsban. And the next best would be a whole group of oils, mostly paraffinic oils or mineral oils. One thing about oils, and this comes again and again in different bioassays, but one thing that was really very obvious when we did those bioassays over the time was that we can see a very nice rate response to the oils. The oils that fruit growers work with, and by the design also grape growers, are usually the maximum field rate is somewhere around 2, 3, 4% in a tank. And the higher the rate we use, as long as it was still close to the label rate, efficacy was pretty decent. It wasn't perfect, but it was better than using some of the other synthetic products that we were trying to test. We test those other products as well, and sometimes we even added some, different adjuvants, and you have the data with new film from Miller Chemical here, and we use a lot of other adjuvants, but it turned out that either chlorpyrifos, Lorsban, or high concentrations of oils are the only effective products. So we learned this in the bioassays that we run spraying eggs directly. So, okay, we have this information, we have this understanding. The next question was, well, how this gonna turn if we try to do it in the field? In 2020, we found a nursery of willow trees where the owner told us, oh, I have such a big problem with the spotted lanternfly. Well, so, okay, we're gonna spray the whole trees. They are tiny trees, as you see, and we ran a lot of different treatments in this particular trial. One thing that we were really interested were, what if we actually use the soap, insecticidal soap, emptied, and then follow it with the oil? Will the impact of the soap help the oil at lower concentration to be more effective? We also tested, a whole bunch of those other oils and so-called organic products and stuff like this. Unfortunately, the results were very, we sprayed, we counted, we evaluated, nothing worked. Even Lorsban, which in every other case was very effective, did not provide any control. So having this behind us, in 2021, working with Brian Walsh, we decided that let's try to give it a commercial try. What I mean by that? Well, we were able to work with the commercial landscaper who actually does it with many other insecticides, who does it normally. So Brian was able to reserve the service of this company. And we found a location, a very nice location in Allentown, Pennsylvania. You have the map of the location and the trees that were individually sprayed on the map in the upper corner. And you see the size of the trees. And we decided that we will prepare the solutions and let the landscaper professional actually do the sprays for us. What we tested? Well, we tested JMS stylet oil at low and high concentration. We tested 3 different concentrations of golden oil. We tested Lesco oil plus Brigade, and Lorsban, and then 2 control. Dry control basically means we did nothing. The reason why we have this strange concentration in treatment number 5, golden oil at 28%, is basically because we ran out of oil. I mean, it turned out that spraying a single tray, and this height, as you see on the photo, takes about 20 gallons of solution, and we still are not able to reach the top of the trays. And we basically, the highest concentration we were able to get with the oil we had on hand was 28%. I know that there is some supplemental label that let you use at much higher rate. So what are the results? Well, the golden oil at 28% provide the best efficacy. Stylet oil or Lesco oil at 3 to 5%, which is kind of really reflection of what we found in all of our lab field bioassays. The survival in the control was anywhere from 70 to 92%. The other columns are, this is how many eggs we've actually tested and how many eggs were per egg mass. Those are some photos from our trials, which I thought would be worth it to share. The first thing is that we only evaluated egg masses that we could reach from those ladders, which, you know, we spend a lot of time, well, not all of us, but some of our people spend a lot of time on the ladders trying to counting those stuff. Also, the highest concentration of golden oil caused a very severe phytotoxicity effect on the tray. At the end, we were actually happy that we could not spray the whole tray because we probably would kill it. This way, we sprayed only the lower part of the tray, and you see how what the difference was between the normal trays and the branches that were treated with the oil. You see this on this photo in the lower right corner or the one in the center. So yes, the higher concentration worked better, but it comes with a lot of other elements. Also, and I think it's also important to say that getting a good coverage on the trays that we work on, and those are not the tallest tray, is basically impossible with the volume of spray that is needed to get the perfect cover. So that's all about the oils. Again, it's a many years of work just trying to present it in 10 minutes. Another piece of work that we spend a lot of time on is really based on our work with the brown marmorated stinkbug, where we use insecticide-treated nets to kill brown marmorated stinkbug adults and nymphs that are attracted to the nets by the attractants, aggregation pheromones that we have available for the insect. We decided, hey, let's do the same with the spotted lanternfly. We use two kinds of nets. We use deterrence nets, which is from Westergaard company, and we use a Mira net, which is from the company from Tanzania. And we expose adults, all different instar of spotted lanternfly for half, 1, 5, 10, 20 minutes, and then put them into the clean container and observe mortality after 24 hours. So this is not how fast they die when they are on the net, but this is mortality after 24 hours after exposure. So in 2019, we ran only adults, and to evaluate, to establish those mortality, we use a probit. And basically what we end up, we were trying to establish the lethal time of exposure that will kill 50% of the population or 90% of population. The zero fly net, the black net, you see the numbers, the exposure of less than 2 minutes resulted in more than 90% of dead adults within 24-hour time frame. For the white nets, it was more than 15 minutes. So in 2020, we actually did this a little bit more. I mean, we knew it working, so we tried to look at different instars and see what we will find out. So we look at the first instars, and then it turned out that, again, the black nets, the zero fly net, the deterrence net, was much more effective than the Mira net. And we basically found out that if the first instar nymphs are exposed, basically walking on the net for less than half a minute, 90% of them will be dead within 24 hours after the exposure. For the white nets, it was more than 21 minutes, less than 21 minutes. Second instar, similar data, slightly longer timing, but again, exposure for less than 20 minutes is causing more than 90% of mortality. The fourth instar and adults, very similar case. Again, if they walk long enough, and in this case was 4 minutes on the black nets, 90% of the population actually was dead within 24 hours. You see relatively low mortality in the control. For the adults, the same case, but we could not actually run the probit analysis for it. So the only thing I can say is that basically you can see that generally adults, most of them were dead with, if they were exposed somewhere between 10 to 20 minutes to the net. But again, I want to stress this out. This mortality was observed at 24 hours after the exposure. What it means is that frequently they actually might be exposed to, they are still capable of flying away, and they, according to our bioassays, would be dead within 24 hours, but they are not necessarily dying when they are on the net. So this is the 2020 data when we use the black net and we actually evaluate separately males and separately females. As expected, and I think this is due to the size. However, this is, I don't have a proof for this. Males were more sensitive to the exposure and the mortality was higher than for females, but there is big size difference between those two genera. So the point here is that exposing spotted lanternfly, adults or nymphs, to the insecticide-treated nets actually can be very effective management tool. However, the question is how we can get them to those nets. I also have in the title of my talk the word exclusion, and this is really the work that was initiated by Heather Rich and my graduate student, Sarah Henderson. She kind of inherited the project when Heather left to Michigan. And we try to look at, can we actually create a wall high enough that will prevent spotted lanternfly from flying into the vineyard? And we use different treatments here. Part of the treatment is we're using treated nets. Part of the treatment is not treated nets and the open space. And Sarah is still collecting this data. We have so far 2-year data on it. We still plan to do it in 2022. And I hope Sarah might be able to maybe share some of her information maybe next year when she will be closer to finalize her research. This is the last slide I believe I have. Well, I want to show this slide because, you know, we love to talk about biological control. And I mean, generalists love spotted lanternfly. Like I said, we don't have spotted lanternfly close to us, but when you go there, when you enough time in those vineyards or woods, you can see a lot of different things actually on spotted lanternflies. The bad part is all of them are generalists, so they are opportunistic. They will take what's come, but they can kill them. So anyway, this is the last slide I have with this. I don't know if I have time for questions or.
Yes, yeah, there's a couple questions in the chat. Thanks, Greg. Okay. For the first question, if I'm reading it correctly, it was, you know, for the first set of results on the ovicidal trial, mineral oil, the high concentration was effective, at controlling spotted lanterns, or at the the eggs. And what was they want to know what the other treatment was that was non-oil that was also most effective.
The only treatment that was not oil that was effective was Lortzman, chlorpyrifos. But since yesterday, chlorpyrifos doesn't have a valid label to use on any food crops. So this is out, it's impossible. Other thing that we tested that provides some efficacy were some neonicotinoids. There was a closer, but we're talking if high concentration of oil, and for me, high concentration is maximum 4%, maximum 5%. When mortality with this concentration of oil was in the range of anywhere from 60 to 80%, any other insecticide that could be used, mortality was in the range of about 40, 50%.
Okay, thank you. And the other question was, I know it wasn't, they want to know if bifenthrin was in the nets, and I know it was not, but if you wanted to sit and repeat again what the two, yeah.
No, those nets are with different pyrethroids. Those nets are commercially produced as a part of the malaria prevention program, mosquito controlling programs, and bifenthrin is not part of it. There is a deltametrin mostly in the deltametrin, and I believe permethrin in the other net.
Okay, thank you. And then just one last one. So were the ovicide efficacy trials conducted in field conditions or in incubators?
We did both. We did both. We did in the field lab where we actually collect them from the field and spray them. But the one that I showed, one on the willow trees and the one on those tall trees in this development, they were just done in the field. We also have the same very similar data when we sprayed with the backpack sprayer, when we sprayed entire vines in the vineyard. Very, very similar results as for the ornamental trees. Okay.
Now I'll get this last one in really quickly, but then I'll have to go to the next speaker. But when treating with golden oil, were the egg masses covered in the oil, or was higher pressure applied to blast off the coating? So they just want to know if there was a difference in the efficacy, if that was the case.
We did golden oil many times, and we try to use golden oil as a direct spray with the handheld applicator, spraying them very directly, and we're not very successful. But we never went with the concentration that was above 5%, because working with fruit growers, I'm really afraid about the phytotoxicity impact. So the one treatment that we went with those very high concentrations, they were done by this commercial applicator, and it was done like everything else. We didn't target egg masses. We're trying to spray the tree to see how this is gonna work. I don't know if this answered the question.
Yeah.
I mean, if there is a specific question in the chat room, I can try to answer them directly to.
Okay.
So thank you.
Oh yeah, thanks, Greg. All right, so we'll go on to our next speaker, which is Dr. Michela Centenari, who's an associate professor of viticulture at Penn State. And if you're all set to go.
Yes. Okay. Can you see my screen?
Yes.
Okay. Let me do this. Okay. So hi everyone. I'm glad to have this opportunity to present some of our work, the work done at Penn State on these invasive pests and how can affect grapevines. So first, I just want to give a few facts about the local, our regional wine industry, because I imagine maybe many of you are not familiar with the PA wine industry. So we have about 14,000 acres of grapes in PA, and most of them, I don't know if you can see my mouse, but most of them are, juice, grapes for juice production, and up here in Erie. But we have more and more wine grapes and wineries in this part of the state. So it's the southeast, south central part of the state. And this is where, unfortunately, the lanternfly was first found. So where we have most of the wineries and the wine grape production. So the lanternfly, at least in vineyards, was first found around 2017. I'm talking about high population of lanternfly in vineyards, and you can see an example here on the left, the picture on your left, with lanternfly completely covering a trunk of a vine. And growers, start to report after a few years of heavy infestation, reduction in yield, and also vine decline. But really, we didn't have any information, or very little information on, how this pest could affect the grapevines from, other countries like South Korea, and also how to manage this pest. But, after 6 years or 5-6 years, now we know much more, right? And growers are able to manage this pest more effectively. So most of our work, at least in my lab, most of our work is done on adult lanternfly. And this is because this is the stage that is most concerning for grape growers. Because they start to see late in the season, like end of August and September, this high population coming into the vineyard. And then this, unfortunately, is when grapes are ripening and when, growers are getting ready to harvest the grapes. So there are challenges related to management, like, what product to use. And plus, we keep seeing this cycle of lanternfly coming back to the vineyard around harvest and even after harvest. From a plant point of view, this is also, a challenging time because this is when, the grapes again are ripening, and the grapes, they need sugar, right, and nutrients to ripe. And when you have lanternfly, especially a high number of lanternfly, they can really ingest and take up a lot of sap, which contains sugars and also nitrogen and other nutrients. So that can be a problem for the ripening of the grapes, but also so late in the season, grapes are start to store again this sugar and nitrogen mostly on the root system, because those are very important, these metabolites for the following year growth. And so it's very important that vines has enough sugar and nitrogen and other nutrients in able to stay healthy, and survive the winter. So this kind of leads to our objective that are kind of mostly, my lab is focusing on understanding how this lanternfly damage the grapevine. And so we hope, with this, we can help our colleagues to provide target management recommendations. And so what I'm going to talk today, most of the focus of this talking is about how lanternfly, again, adult lanternfly, can affect concentration of important metabolites for the grapes, carbohydrates and also nitrogen. And also, how lanternfly, if and how, is able to affect not just the accumulation of this compound in the vine, but also the assimilation, so the photosynthesis. And again, because those metabolites are very important for the grapevines to stay healthy and to produce good quality grapes. And also, we are interested in knowing if there is a population density response. So in other words, if the vines are able to, tolerate a low population, and this, we start to see maybe negative effect only when we have medium or high population density. So this again will help to provide targeted recommendation for management. So I'm also going to quickly summarize 2 studies that we already completed and we hope to publish very, very soon. So the first was done on field-grown vines, 5-year-old Riesling, and was done at the Grower Cooperator, and we caged 16 vines for 2 years. And every year we use different vines. So you can see a picture of some of the experimental vines. So each vine has its own, a little cage. And basically what we did, again, late in the season, starting mid-August or late August, we start to introduce lanternfly on these vines, and each vine got a different number of lanternfly. So we wanted to go from like very low or 0 lanternfly to very high number of lanternfly. And I put the rate, so the density down at the bottom of the slide. And we also want to mention that we introduced the lanternfly in cycles, try to mimic what growers were experiencing. So basically we introduced the lanternfly and let them on the vines for like 5 days. We removed them by hand, and after a few days, we introduced more lanternfly. And this, we did this from August until after harvest, October. And in 2020, to confirm some of these, the results that were obtained with the field-grown vines, we did also a second study on potted vines, Marquette vines. And in this case, we only, had 2 treatments. We had the control, no lanternfly, and other vines, 6 vines, half of the vines were assigned to a high population density. And so we basically infested those vines with 80 lanternfly per vines, which is considered high number because those are potted vines, so are smaller than the field-grown vines. So this slide kind of summarize our results obtained at the end of the season for the carbohydrates. And this is mostly the work done by our student, PhD student, Drew Harner. And I want to specify that for the carbohydrates, we mostly specifically look at the starch, because starch is the storage carbohydrates, what the vines use for storing the sugar in the fall and to grow basically in the following year. So it's a very, very important sugar. And also we look, of course, of soluble sugars. And basically the colors, on this slide, what they represent, the red represent if we see a decrease of these compounds, of these carbohydrates with increased number of lanternfly. So a negative effect caused by lanternfly, and the green is the opposite. So definitely the most striking, I guess, the most important results was this drastic reduction in starch in the root system of the vines. And this, for 2 years and also in both experiments. But also, what was interesting that we also see a different allocation of these carbohydrates. For example, the starch, we saw, a strike reduction in the root system, but at the same time, on the same vines, we saw an increase in starch in the canes or in the stem when lanternfly were feeding on them. And also we saw that while the starch was decreased in the root system, the soluble sugar slightly increased. And unfortunately also we saw some effect on the fruit at harvest, some reduction in the sugar at harvest. So I'm going to show you some graphs, of these results, hopefully, to better, so you can better understand, I guess, our finding. And I'll show you only the 2020, because basically we obtained very, very similar funding in 2019. So this is the root system, the data for the root system of starch. And we collected the roots, these woody roots, at the end of the season. So after leaf fall, or close to leaf fall. And basically each of these dots, so we have 16 black dots, each one represent one vine. So we go from like 0 lanternfly, so vines with 0 lanternfly, and moving to your right, we have vines with more and more lanternfly. This one at the bottom of vines with, we standardize by shoot, so it will be 15 per shoot, which approximately is 200 per vine. And you can see that there is really, a significant reduction in starch in the root system. And mostly this reduction is very, very significant when we have a high number, like 10 or more than 8 lanternfly per shoot. And when we compare those values, to what has already been published in the literature for other even fruit crops, and which have been exposed to stress or like biotic or abiotic, we definitely found that those values, like 15, 20 milligrams per gram of tissue, are considered very, very low, are considered like value that can compromise the fruit crop or the grapevine health. So this is kind of concerning, but also I wrote down there that these vines were exposed to lanternfly for a total of the 28 days, which is, a very extensive feeding. And hopefully, I'm sure the growers will never let the lanternfly feed on the vines for 28 days. But we are trying to push those vines to extreme, just, to better understanding how the insect will, affect the vines if it is not managed. And quickly I can show you the results for the second trial for the potted vines. Still the starch in the root system at the end of the season. And again, we found that vines exposed to lanternfly for an extensive period, 40 days, had a 77 less starch than the control, which one, the vines with no lanternfly. And at the same time, as I mentioned earlier, we found that the same roots, they have increased soluble sugar. So we're now, we want to investigate what's going on with the vines. We want to know if the vines are kind of converting starch in soluble sugar and moving the soluble sugar up to the above ground tissues where the lanternfly are feeding, because we saw this increase in starch on the stems. And this is, some opening question that we are investigating. But unfortunately, this strong reduction in starch can again compromise vine health. And I want to mention that the spring following the lanternfly infestation, we found that 2 of the vines that were assigned to the lanternfly treatment were dead. I mean, there was 0 bud survival on those vines. So it was concerning. And again, we pushed those vines to extreme. And also quickly, I wanted to mention the results for the above ground tissues. And this is the starch measured at the end of the growing season, after the vine have been exposed to lanternfly for a long, 28 days. And again, what was interesting is that in this case, the starch, both in the bark include the phloem, and in the wood, which is the interior part of the cane, which include the xylem. In both cases, the starch is increasing. So more lanternfly, more starch on those tissues at the end of the season. And that is where most of the lanternfly were feeding directly on the stems. So, summarizing below, we saw the strong reduction in starch in the root system, but at the same time, an accumulation, increase in starch in the stems. And last, for the starch, for the carbohydrates, I also wanted to show the total soluble sugar measured at harvest on those vines. We saw a slight reduction. This is a white variety Riesling, so usually we don't, get very high sugar at harvest. And we saw a slight reduction, definitely was more like relevant again when we have very, very high number of lanternfly, but was not as relevant as the starch in the root system. Briefly about the nitrogen, again, we measure nitrogen in the same tissues at the end of the season, but we did find a decrease in nitrogen in the root system and also in the leaf blade. No effect on the stems or on other organs, but this reduction were not really so significant like those for the starch, and they were not in what we call a deficiency threshold or range. But this is only one season of infestation or subfeeding. Things can change probably, if the lanternfly keep feeding on the same vine year after year. And last, also one more graph. I also wanted to talk about the photosynthesis, because I mentioned the effects on the accumulation of carbohydrates, but also we wanted to check or to investigate if these pests can affect the ability of the vine to assimilate, to produce basically sugar and assimilate carbon. And so this graph is divided in 3 sections. So basically these show what, these measurements were done or conducted 6 days after lanternfly were feeding on those vines. And you can see this, data points are spread, all over. Again, each point is a vine. So we didn't see a strong correlation, a strong reduction in carbon assimilation due to lanternfly, except these 2 points on the right when there was, very high number of lanternfly on the vine. But things change, 2 weeks later, 20 days of lanternfly feeding on the vine. At this point, you can see the slope going down, sharp decline with increasing number of lanternflies. So definitely we saw a strong effect of lanternfly in reducing carbon assimilation after extensive feeding, 20 days or even 28 days. And as I summarize at the bottom, if lanternfly keep feeding on the vines and they're not managed, definitely, at least in for this experiment, we found a reduction in carbon assimilation. So to summarize, those results, again, we know growers are managing lanternfly, and it's good to keep them away from the vineyard. But if they are on the vines and extensively for several weeks, they can really decrease the vine ability to produce sugars and also the accumulation of these important metabolites at the end of the season, and most importantly, the starch. So overall, we consider a lanternfly a stressor, like another additional stress that the vines has to deal with, in addition to other abiotic and biotic stressor. So unfortunately, it's something that the growers needs to deal with, in addition to other issues. And what I show you are results from one vineyard and a potted vine experiment. But definitely, response of vines can change. Or we think can change depending on not only population density, but also on the baseline health of the vines before being infested with lanternfly. Other factors can change vine response or vine age, and definitely the presence of other stressor, environmental and pathogen stressor. I just quickly want to mention that right now we are still analyzing all the data collected in 2021. And one thing we really actively work on is to understand how the feeding affect also the wine, the composition of wine, both red and white variety. We are also trying to better understand how the carbon is partitioning between tissue and organs in response to lanternfly feeding. And we are, again, still analyzing the data for that experiment. And also we are starting a new study with Flora Sivido, our entomologist here at Penn State, to, again, better understand if cumulative effect year after year can really, impact in more relevant ways also the vines. What happens if, lanternfly come back to the same vineyard year after year? And so with this, I'm done. I hope I was within the 20 minutes. And definitely, I want to thank everyone, all grower cooperators first, but also all, the team, all the people at Penn State that make this work possible. And Greg was mentioning, we are now in the quarantine area. And for us, actually, our experimental sites are usually 3 hours from Central PA, where we are. So, there's a lot of travel and teamwork to make these experiments successful. So thanks for your attention. And I don't know if I have time for any questions.
Yeah, thanks. That was excellent. There was just one question. Did you follow up on carbon assimilation rates later on to see if they rebounded at all?
No, I mean, the first year I mentioned like we did the cycles, right? So we measured also photosynthesis in between the cycles where the vines didn't have any insect, and we didn't see any recovery. We didn't see like the, down, up, like this kind of recovery. They were just slowly going down. Yeah, that's a good question. Unfortunately, we didn't see a recovery in our study.
Okay. Thank you.
Thanks.
All right, so we'll move on to the next presenter then. Dr. Cristina Rosa is an associate professor of plant pathology and environmental microbiology at Penn State, and she'll be talking on grapevine yellows.
Yes, so actually today, can you hear me okay?
Yes, yeah.
Yeah, good. So I'm talking actually about a few basically parts of our research. So I strictly collaborate with Michela Centinari and with Julie Urban for these studies. So my lab doesn't have an extension component, so I rely on them to do field work and insect work. So what I'm going to present is some plant responses due to high spotted lanternfly density infestation, and then the capacity for the spotted lanternfly to transmit pathogens. And of course, I put here on the first slide the funding agency because they are the same as Michaela mentioned. So USDA, so basically PDA and Wine Marketing Board. Okay, so for the first part of my talk, I'm going to link this to the potted experiment that Michaela just showed, where she had 12 potted 7-year-old Marquette vines that were caged. And if you remember, she used 6 with 80 spotted lanternfly adults for 45 days, and then 6 were left as a control without insects. And so at the end of the 45 days, she gave us canes, and we did RNA extractions on those canes. That is the site, as Michaela said, where the spotted lanternfly adults feed the most. So we conducted a unique dual index library for each sample, and then we send this sample for RNA transcriptomics. So we got very good coverage for 58 million reads per sample that we got. And so one of my students, Tarek, he cleaned the reads, aligned and assembled them. As a reference genome, we use Pinot Noir, which is one of the basically ancestor, let's say, or the parental lines for the vines. And we also basically had to do a lot of work with Arabidopsis because the Pinot Noir genome is not so well annotated. So I wanted to present the results that we have seen in terms of what a spotted lanternfly does to heavily infected grapes. So what you see here in this graph is we looked at how closely related are all the vines that were fed on by the spotted lanternfly at a heavy number compared to the one that were not fed upon. So if you look at the left, you can see this distance matrix, and you can see that the sample groups very well together by treatment. So you have the control, they are the C, and the spotted lanternfly infested plants are the S. So also we did a kind of PCA analysis to explain the variation of the gene upregulation and downregulation that we were seeing in our sample. And again, you can see that all the green dots, I don't know if you see my cursor, on the left are the control, and they cluster very well together, and all the spotted lanternfly cluster on the right. And so we can explain the distance between the control group and the spotted lanternfly group is about 83% of the variation. And then among the different samples, we have only 6.2% of the variation. So this is a volcano plot where for each of the single dots that you see in this graph, this is a gene that has been upregulated on the green or downregulated by our spotted lanternfly treatment compared to the controls. And then the dots that you see in black here are the gene that they were upregulated or downregulated, but not in a statistically significant manner. So what we found was about 42,000 genes were regulated by spotted lanternfly, and 4,793 were statistically differentially expressed, and we have about 3,400 upregulated and 1,200 downregulated. So a lot of genes. So the spotted lanternfly effect is pretty, at least a heavy infestation, is pretty well felt by the plant. At this point, what we did is we had to compare our gene identities and figure out what these genes do. And so what we did is looking at the gene IDs, both in Arabidopsis and grapes, and try to see what unique genes and function we can analyze. And then we have to reduce the redundant analysis. So basically, there's a lot of manual curation. But basically, this graph on the left, the A, shows basically the molecular functions of these genes. And so we group them in biological pathways, molecular function, cellular component, and KEX pathways. And the colors represent the different statistical, basically a fast discovery rate, if these genes are really upregulated or downregulated, and the size of the bubble represents how many genes were upregulated or downregulated in that category. On the right is just the significant grouping functional categories. So we would have defenses, phytohormone signaling, metabolism and signaling, transcription factor, transmembrane, and so on and so forth. Okay, so this is the first screen that we have for the transcriptomic, and I'm going to show you some categories of these biosynthetic pathways that they were interestingly regulated, and just let you know what genes we found that they were interestingly grouped. So the first category that you see here grouped in this graph, so there are differential genes regulated in the category as plant responses and plant defenses. The spotted lanternfly triggers a very high defense responses pathway, and these ones are pretty generic. So you see a lot of oxidation reduction processes here, leaf senescence, defense responses, and also response to abiotic stresses, response to cold, some response to herbivory, and also metabolite biosynthesis. So all the major classes of oxyreductases family were upregulated here. And we think that the main responses in plants in this case are in the antioxidant pathway under the spotted lanternfly treatment. So peroxidases and lacases were also very enriched together with reductases, transferases, some cytochrome P450. So these are all genes that are not only involved in regulation of plant defenses, but also stem lignification and stomatal closure, which are typically seen when you have a heavy herbivory. There is also a lot of homeostasis and stress signaling going on here. So then I proceed to the next pathway. So these one are differential gene upregulating and belonging to regulatory pathway for adaptive immunity and growth. So on the left here, you have a hormonal pathway. So these would be more adaptive responses of the plant defenses. And interestingly, we didn't see so much differences between the salicylic acid and jasmosic acid hormonal pathway transcription regulations, but we've seen more in the abscisic acid pathway. So abscisic acid, of course, is very important for growth of plants, but also for plant defenses. On the right-hand side here, you see also all the other like transcription factor that we found. So transcription factor are hubs for regulation of plants. So they are acting in the nucleus and they are starting all the other pathways. So again, these show that the regulation of the spotted lanternfly upon the feeding on the plant is heavy. I can give you some, for instance, this MIB transcription factor is a family of proteins that contains the highest number of genes and are always found in cellular morphogenesis, but they are also important for secondary metabolism, meristem formation, and plant stress responses. What is also very important is to see what genes are basically included in multiple pathways. And a way to do this is looking at what transcription factor can regulate pathways that are commonly up and down regulated. And so this graph here is looking at genes in the defense pathway, phytohormone pathways, and transcription factors, and is looking at what genes are in common between these classes. And so you can see, for instance, that the defense pathways, they have 862 genes upregulated and downregulated, the phytohormone 100, and then in common there were 136 genes also with the transcription factor. So we think that this transcription factor is actively acting upon starting these regulation defenses for phytohormone and primary defenses. Okay, so going down toward the list, we also saw many metabolic processes, signaling and photosynthesis that were regulated. Interestingly, we saw upregulation in photosynthetic genes, and we don't know if it's upregulation of those photosynthetic genes in the stem has anything to do with the regulation that Michaela saw afterwards in the canopy. But anyway, that is very interesting that we saw photosynthesis, response to light stimulus, light harvesting, photosynthesis, and so on, genes upregulated. We also see a lot of microtubule and cellular factors that they are involved in transport of signal that they are upregulated by the spotted lanternfly. And then here on the top, you can see metabolic processes, carbohydrate, for instance, response to sucrose and starch catabolic processes were also regulated, maybe signifying that the feeding for the spotted lanternfly induces plant to repartitions and to change kind of the way of how they are producing sugar, starch, etc. Since I gave a lot of names and a lot of pathways, I wanted to give an overview of what we have seen. So if you look at these kind of cartoons, we are showing here how spotted lanternfly could act on the cellular level on grapes that are heavily infested by these insects. And so the spotted lanternfly would damage the plant, and so the plant would respond by fortifying and remodeling their cell wall. And cell wall remodeling brings to a lot of reactive oxygen species production antioxidant, and is also translating into a lot of activation of plant defenses through these protein kinases. The signal enters into the nucleus of the cell. You have a remodeling of basically the chromatin structure, and you have transcription factors that now are getting activated. And this transcription activator brings to calcium signaling, phytohormone production, primary metabolite production, photosynthesis production, and secondary metabolite. One of the category of genes that we have seen that was most regulated has to do with increased callus deposition and stomatal closure, and so signifying that the plant is trying really to resist to the spotted lanternfly, but the spotted lanternfly probably has the, is still causing a lot of damage to the plant. Okay, very briefly, I will go to my second objective, which is many growers are really worried about, are the spotted lanternfly able to vector pathogens? And I'm a plant virologist, and I've worked with insects that they are sometimes transmitted different type of pathogen. And so what we focus in my lab in this case was looking at the specific ability for the spotted lanternfly to transmit Pierce disease, North American grape yellows, and grapevine red blotch associated viruses. And I will explain you why in the next couple of slides. So we selected Pierce disease of grapevine because even though Pierce disease is not very important here in the eastern United States, it's a very important problem in California. And who knows if the spotted lanternfly is going to arrive there or other warmer places where Pierce disease is a problem. So the causal agent of Pierce disease is Xylella fastidiosa, and it causes a major problem in basically not only in California but also in every other growing region where the disease is present. It's transmitted by spittle bugs and sharpshooter leafhopper. And what is very important to notice is that the problems seen with the Pierce disease in grapes is amplified when you have very robust vector, such in California for the Homalodisca vitripennis, which is the glassy wing sharpshooter. We also were interested in looking at the possibility of transmission for phytoplasma, which is another group of bacteria that are, in this case, wall-less. There are many phytoplasma causing the same disease, which is North American grapevine yellow. This is a common disease seen here also in the United States in our region. And the pathogen is transmitted by phloem-feeding insects in the family Cicadelidae. Again, this disease is very severe. It can cause grape cluster shriveling, lignification of canes, dieback of canes, and plant cell death. The last pathogen we were looking at is grapevine red blotch associated viruses. This is a pretty common virus, but has been discovered recently. So we don't know that much about the vector of this virus. We know that it is vectored by the three-corner alfalfa hopper. And again, it is a pathogen that causes reduction in fruit quality, ripening of grape usually is delayed. And so you have also lower crop value and loss of marketability. So why were we looking at, in particular, at these grape diseases? There are tons of them out there, but it's because all the vectors that transmit these diseases are closely related to the spotted lanternfly. So you can see these phylogenetic D3 here, and I highlighted in red the vectors of the diseases I mentioned before, so spittlebugs, leafhopper, plant hopper, and then where our spotted lanternfly is. So there is actually the possibility that spotted lanternfly could serve as a pathogen vector. So our experiment, so we take spotted lanternfly, 4 instar or adults, because those are, as Michaela mentioned, the insect that they are most seen in our region in grapes toward the end of the season. And then what we do, we put them on infected grapes, move them to healthy grapes, and then we test afterwards these healthy grapes to see if there was a pathogen transmission. We already did some experiments last year and the year before. So we use around 60-40 instars or adult spotted lanternfly, and we left them 72 hours to feed on infected plants, moved them to uninfected plants for 21 days. After 21 days, we take the survival individual and test them to see if they still retain the pathogens we are looking for. And then we let the plant develop a little bit more. Maybe we test them in following seasons, and we look at look to see at the molecular level if the plants, the recipient plants became infected or not. So I just give you briefly the first results that we have. So we did a 1-season experiment with grape and red blotch associated virus, and we didn't get any transmission. Doesn't mean that that cannot occur, but we don't think that is very likely that the spotted lanternfly would transmit this pathogen. So far we tested 11 plants that we use as a recipient plants, and also the insects that were collected after the transmission experiments, and none of the insects resulted positive for this virus in none of the plants. So we also tested already transmission for grape and yellow phytoplasmas, and out of 16 plants that we tested for transmission, one of them was fed on upon 4 instars nymphs that they were left feeding on infected plants with phytoplasma, and one of these plants became infected by phytoplasma. This plant was in a cage, but we still are not 100% sure, even though we buy certified grapes that they are supposed to be phytoplasma free, we are not 100% sure if this result is correct. We send we are performing now the analysis of the second trial to see if we can replicate these results and confirm that actually the spotted lanternfly could be a vector for phytoplasma. We will start disease transmission trials in fall 2022. Okay, so one of the things Michaela mentioned is that grapes are exposed to a variety of pathogens and stressors. And so we wanted to know if the spotted lanternfly responses we see at the transcriptomic level are shared by grapes that they are infested by other, for instance, hoppers, or by, for instance, Pierce disease. And we want to know if the responses that we see at the transcriptomic level can be used to predict the health of plants once they have a spotted lanternfly and maybe other pathogens. And so we did an analysis here where the Marquette data are our data for at the transcriptomic level. So we look at the number of genes that they were upregulated similarly in our experiments. And then we use another transcriptomic data available, and this was generated on Chardonnay and Moscato Bianco. There are 2 different varieties. Chardonnay is more resistant to phytoplasma than Moscato Bianco. And we also know that these experiments were done with hoppers, and so we are looking at the responses of these 3 basically grape variety upon feeding either by the spotted lanternfly or the vector of these phytoplasmas. And so we can see that there are a lot of genes that are similarly regulated and also differentially regulated in the 3 varieties. We also look at the Pierce disease up and down regulations. This study was performed at UC Davis, and the transcriptomic is available. And so we saw that 600 genes were similarly regulated by spotted lanternfly and generally regulated by Pierce disease. So these are the categories here that are commonly regulated by Chardonnay. So you have defense responses, cell wall organization, protein phosphorylations. So they're all pathways that are generally induced when you have a heavy damage. So why we want to do that, again, is because in the future, we would like to see the interaction of pathogen and spotted lanternfly on plant health. One of my students, that is Michaela, is also quite biasly, Crossley, is also trying to look at the preference of spotted lanternfly for healthy versus infected plants, and to try to see if growers should care if they have an infection of pathogens plus spotted lanternfly in their vineyard more than if they have one or the other. So thank you for your attention. If there is any time, I will take some questions. I will stop sharing.
Okay, thanks, Christina. That was great. Yeah,
Thank
there's just one question, but if I could ask you to answer that like offline, so we could get on to I can move on to the next presenter to make sure
Sure.
she's got enough time to get her presentation in. All right,
Mhm.
thanks. Okay, so our next presenter then is Dr. Ann Nielsen from Rutgers University, and she's going to be talking about movement of SLF in vineyards.
Thank you, Greg.
Yep.
And thank you. Good afternoon, everyone, for sticking around for the Spotted Lanternfly Summit. Okay, well, thank you, everybody. I'm going to be talking today about Spotted Lanternfly movement in New Jersey grapevines. And as the only presenter from New Jersey, I wanted to just give a quick overview of what's going on here. We have had spotted lanternfly. It was first detected in 2018 in northern New Jersey. The first detections were kind of exactly where we would predict them, and they were right over the river from our neighboring state of Pennsylvania. And so that's where our populations first started off. The New Jersey Department of Agriculture immediately put in place a 3-county quarantine. That was expanded the next year to 8 counties, all again bordering Pennsylvania, and then this year it was extended into a 13-county quarantine. As of the beginning of this year, last year, as of 2021, we had the New Jersey Department of Agriculture as well as Rutgers University Cooperative Extension had a community science reporting component that allowed us to track the kind of detection or observations of spotted lanternfly throughout the state. It has been detected in every single county except for Cape May County, which is our southernmost county in New Jersey as of this year. Population densities and impact vary greatly between all of those counties. This is a picture from Warren County, and I love this picture. This was taken by my student, Kasia Matalinska. She's a PhD student in my lab working on spotted lanternfly, and she conducted or assisted with almost all of the research I'll be presenting today. But this is a population of spotted lanternfly on sycamore, and this was at a grape vineyard in northern New Jersey. And we collected, I think, 3,000 bugs from this plant in a matter of, as fast as we could collect them. So sycamore is a really important host, in addition to the black walnut and tree of heaven that we also see. So we've been collaborating with the USDA lab in Kearneysville, West Virginia, led by Tracy Lesky, on using a circle trap to intercept populations of spotted lanternfly as they move along. This trap encircles the tree trunk and basically captures them as they move up and down the tree. It's very effective for the young instars, and I want to thank Greg Krawczyk for putting up a slide of the life cycle of spotted lanternfly, so I could remove that from my talk. And one thing, what I really want you to take home from this slide here is that in 2020, we had really high populations. Again, this is primarily first and second instars of spotted lanternfly. And it looks like our populations were really high in 2020, and then they dropped in 2021, but that's not exactly what happened. We actually had more bugs in 2021, but that's because our sampling increased. So this is, if you look at the mean per trap, that's why this happens. And if we break this down between northern sites and southern sites, what we see is a switch. And I think this has happened in Pennsylvania as well. I kind of call it an invasion donut effect, where we have our first initial population of spotted lanternfly, which for us occurred in northern New Jersey. Our populations were really, really high in those locations, and that's where our trapping primarily was centered in 2020. However, in 2021, we expanded our sites. We were able to trap throughout a larger area of New Jersey because it was present in larger numbers throughout the state of New Jersey. And we trapped on multiple host plants, expanding beyond tree of heaven and black walnut. And so what we saw was a switch where now our populations in southern New Jersey are much higher, and in some spots in northern New Jersey, we're finding very few spotted lanternfly. But what I'm going to be focusing on today, so this year, what we're seeing, I mentioned already, an expanded quarantine to 13 counties. And that's just really representative of the larger impact and spread that spotted lanternfly is having throughout the state. We're seeing a lot of community questions on spotted lanternfly and management within the landscape, and questions from landscape professionals. So we're not really seeing populations in large numbers, or at least in damaging numbers, in farms other than vineyards. And that's kind of consistent with what we would expect. But we are seeing massive problems in many New Jersey vineyards. Again, these are just some pictures. You've seen very similar pictures. We're seeing egg masses, we're seeing early instars, and we're seeing large populations of adults. Now, we did do a survey of our growers both in 2020 and 2021. And we've also done some surveys for egg masses in woods adjacent to vineyards. And this was work done by my graduate student, Kasia Matalinska. What we did is we surveyed trees in the woods along transects going from the wood edge into the woods. And then we surveyed the plants that were surrounding them. And what we found was that 85% of the egg masses that we found in wooded areas adjacent to vineyards, 85% of those were within close proximity to wild grapevines. And so at least early in the invasion stages, this work was done in the winter of 2019, 2020. So that was within after our first full year of having spotted lanternfly in the state. So wild grapevines, if you're doing surveys in new areas, in wooded areas, wild grapevines, at least for us in New Jersey, were really indicative of having spotted lanternfly egg masses on trees surrounding them. We did see a 2-year delay between the first identification of spotted lanternfly in those wooded areas and problematic numbers in adjacent vineyards. And when I say problematic, that varies a little bit depending on the grower that you're working with, but I'm talking about at levels of 10 on average per vine, where I would suggest a management tactic would be needed. So grape is a very suitable host for not only for egg masses, but also for early instar spotted lanternfly, and I'll get to that in a second. But we did conduct a survey of our New Jersey growers. We didn't have great response, but we conducted a survey in 2020, and about 10% of our New Jersey wine grape growers had spotted lanternfly. It's a little different than our survey, our own surveys through our IPM program, where we estimate about a third of our growers had spotted lanternfly. But of the growers we surveyed, even though only 10% of them said they had spotted lanternfly, 42% of them had applied insecticides as a preventative measure for spotted lanternfly. In 2021, those numbers shifted, and 80% of the survey respondents in New Jersey indicated that they had spotted lanternfly in their vineyard, and 80% of those growers had applied insecticides. So essentially, we eliminated the kind of scary the applications for insecticide just out of fear, and if they had spotted lanternfly, they were applying insecticides. And this resulted in between 1 and 9 extra insecticide applications targeting spotted lanternfly alone. So Kasia did some research trying to figure out if grape was a suitable host plant for development of spotted lanternfly. And what we did this work in 2020 and 2021. And we include multiple host plants, specifically known host plants such as black walnut, tree of heaven, red maple. And then we used early grapevines that we had established ourselves. And then in 2021, we added in some established grapevines because essentially the nymphs sucked the life out of those newly planted grapevines. And what we did is we caged individual instars. So, 10 first instars on a plant, 10 second instars. And then we also did third and fourth instars at slightly lower densities with between 8 and 16 replicates per life stage per host. And the point of this was to look at by life stage, which host plants we can target surveillance on and maybe even management, and how each host plant impacts survivorship and development of spotted lanternfly. So these graphs are a little busy, but what I wanted to show you was that for the first instars, we have black walnut here in black, grape in purple, and grape in yellow, red maple in red, and tree of heaven in green. And so grape and tree of heaven and even black walnut were really suitable for spotted lanternfly growth. Their development time was not significantly different from each other. However, red maple, it looks like they can survive on red maple in the first instar, but they they didn't really complete development. So the development time is really, really long. For second instars, we don't see any significant difference between our host plants. And then when we get to third and fourth instar, we start to see differences. Red maple drops out. It's not a suitable host plant anymore for third or fourth instars. And for our fourth instar, our grape plants really do not support, they don't support development as well. And we can synthesize that all by looking at a suitability index, which is looking at the average time for development and percent survival. These numbers here in bold and blue indicate that they were highly suitable for spotted lanternfly development. And the numbers in red indicate that they're not. So in terms of grapes, we know that black walnut is a really important host plant. We know that tree of heaven is a really important host plant. But grapes are also emerging as a really important host plant, especially established grapevines for first and second instars. And they're not as well supported for fourth instars. And that's kind of reflected in our phenology data, which I'll show you in a second. So going back in time a little bit, in 2020, we did some surveys at 3 different sites. These are all commercial vineyards. So like our other speakers other researchers working with spotted lanternfly and grapes, my research station is located down here. And so we have to travel throughout the state, sometimes up to 3 hours away to get to our sites. We have to work where the spotted lanternfly are. We did not have spotted lanternfly at my research farm here until late last season. So we work with commercial growers who are trying to allow us to do the research, but also trying to manage and protect their grapevines as well. And so it's a big dichotomy, but it creates really strong relationships as well. So the population up here in Warren County, we had really, really high numbers of spotted lanternfly. We take our data on a panel of grapevines, which is about 6 grapevines. And we had upwards of 800 spotted lanternfly in those 6 vines at certain time points in the season. But we didn't really see spotted lanternfly moving in until right after Labor Day. Populations were also high in Hunterdon County. It doesn't look like they're high just because of the scale from the Warren County site, but we definitely exceeded that 10 per vine threshold that they use in South Korea. And we had very, very few numbers down in Salem County. We did survey at these same vineyards in 2020 for the nymphs, and we only really found them at this Warren County site. There's 1 or 2 nymphs found at the other sites. But this year, in 2021, we conducted our surveys at 5 different vineyards throughout the state, and we see really drastic numbers. So we have high populations of first instars, second instars, third instars, and the third instars decline. This gray bar here is at a farm that did not manage for spotted lanternfly, and so the populations are much higher, reaching a peak of third instars at 14 per vine. I don't have fourth instars on here because we really didn't see any. And then of course our populations of adults vary quite a bit. They're coming in right around Labor Day as well. We do see some populations moving in mid-August, and most of the variation we see there is due to insecticide application. So the nymphs are quite abundant, and we're recommending management against the nymphs to time with either great berry moth application management or Japanese beetle management, preferably great berry moth because the timing is earlier for the first and second instars. Now, I mentioned we saw in our trapping network with the circle traps, we saw a switch from our populations being high in North Jersey to the populations being high in South Jersey. We saw the same exact thing with our vineyard surveys, to the point where we saw no adults in spotted lanternfly in the vineyard in Warren County. They did eventually show up after we stopped surveying, but the populations were much lower, whereas the populations in Hunterdon County were really high. And this is with aggressive management, which I will get to in a little bit. And then the populations in Salem County, which in 2020 did not really have any, were also required 1 or 2 management applications. So we wanted to look to see looking at this movement of spotted lanternfly into the vineyard, specifically of the adults. You know, the adults are coming in, they're feeding heavily, they're, you know, as Michaela mentioned already, they're causing damage to the plant by removing essential carbohydrates and sugars. So why are they moving, and how can we, is there time component to that movement that we can then integrate into our management categories? So we did this in a couple different ways. We started off by wondering if we can intercept this movement into the vineyards. And then this year we looked at characterizing that movement into the vineyards. And we do a lot of mark release recapture work in my lab. And for spotted lanternfly, we mark them with fluorescent powders. And then we go back at night and watch for the glowing bugs, which is kind of fun. So we also looked at the Vestagard netting as a management tool. We applied it a little differently than Penn State did. Vestagard netting is impregnated netting that's used for malaria control and is pretty effective at controlling spotted lanternfly in when it comes in close contact with it. So we wanted to look to see if we could provide an affordable solution for spotted lanternfly management by intercepting the movement of the adults from the wood edge into the vineyard. And we tried different heights. We tried it at low, which is a ground deployed treatment, mid canopy covering the fruiting zone, and then an upper canopy covering the top zone. So each of these sections is about 2 feet in height. And we did this across 25 feet, 25 or 50 feet, I think it was. So essentially, we see the bugs coming in. We had a little lip in the netting to catch all the dead bugs. We counted dead bugs, we counted live bugs, and then we released a whole bunch of marked bugs to see if we could actually intercept their movement. And what it comes down to is, unfortunately, mortality is not any different than the grower insecticide management treatments. This was a very heavily managed vineyard. But when we look at the bugs that we release that are fluorescently marked, we do see that low deployed netting that's 2 feet from the ground, or ground to 2 feet up in the canopy, did cause the highest percent mortality of the bugs that we released. And most importantly, the bugs that we released, the netting does intercept the movement. So the netting prevents the bugs from moving further into the vineyard than the border, which is really effective. And then, a portion of those are controlled. It was not as effective as we were hoping to. And when we tried it again this year at a different farm, because the initial farm had no populations, obviously we killed them all before. This was not very effective when that distance from the wood edge was reduced. So we're not pursuing the insecticide netting as a barrier treatment anymore. It's a little tricky as an affordable insecticide management tool. So our second question here was, can we characterize the movement into the vineyards? So again, we marked these bugs with fluorescent markers. And by doing this, we actually marked them in situ. So we'd see the bug on the plant during the day and we'd puff fluorescent dust on them. We also were covered in fluorescent powder. So it was a quite colorful event. Each week had a different powder. And if you see here, there's some bugs that are dead on the ground. They're both marked with yellow fluorescent powder and pink fluorescent powder. So each week we counted the number of live bugs. We also counted the number of dead bugs, which was a back-breaking activity, and the number of those bugs that were marked. We did that at night to visualize them. And then to kind of what we were trying to do was to not only calculate the population size, but to calculate out when the bugs are moving in, how long they're staying in the vineyard, and how effective these management tactics are against them. So we did this.
Can I just check with you real quick, Anne, on it? Like, if you have very many slides.
Yep, I have like 4 more slides.
Okay, thanks.
So what we see here is we see a strong increase in both sites at Labor Day, that September 9th time point. And these declines that you see are due to grower management applications. And here's an indication of how effective those management applications are, because we counted all those dead bugs. When we apply Mustang Max, we get pretty good mortality. Same thing with Octara. And then that grower just alternated between those two. So what we see here is that Mustang Max is about effective for 3 days, whereas Octara was effective for at least 10. And that's kind of shown here as well with the total number of bugs and the number that were killed for each of those management tactics. But what's really interesting about this data that I think is that the bugs are persisting on the vines for about 3 weeks. So the bugs that we're marking, we're able to recapture them. We see a lot of immigration and emigration moving in and out of the vineyard, but 24% of those bugs are staying in the vineyard, and they're staying for 2 to 3 weeks, even with insecticide applications. So I think that that's really encouraging. And the populations that we're seeing, of course, are just a small fraction of what we're able to capture. So, and we did recapture 24% of the bugs, which is really impressive for any mark release recapture. So what we know now about spotted lanternflies in vineyards, at least in New Jersey, is that the peak immigration occurs in the first week in September, and we've seen that for 2 years at multiple sites. This may be related to nutrient declines in the host plants in the surrounding woods, but we also see an increase in the fat content in those females that are coming into the vineyards, those yellow bellies, when they're full of fat bodies. And they can persist on the vines for 2 to 3 weeks. And again, our Arctara is causing 100% mortality on day 3 and up to 80% by day 10, whereas Mustang Max is causing about 80% mortality on day 3. So in New Jersey, this is a vineyard here. You can see all these egg masses on this vine, this post. This is the first year that they found spotted lanternfly in this vineyard. So we have definite problems with spotted lanternfly in our vineyards, but we have a lot of tools to control them. So sorry for going a little bit.
I know, it's okay. Thanks, Dan. Yeah, that was really good information. That's why I didn't want to hurry you or rush you off if I could prevent it.
No
But
problem.
yeah, all right. But yeah, you probably have to check the chat or the Q&A to see if there's questions for you. But yeah, I'll turn it back over to Jay for the next speakers then. And thanks for all the presenters, the 4 presenters that presented on their research today.